Okay, so we began discussing human tissues, and we said that tissues are aggregates, they are collections, they are united cells. So these cells, they attach to each other. They are glued to one another. The cells in the classroom right now, you are separated from one another. You're not touching each other. But the cells, they are like in the auditorium. In the auditorium, people are still not touching each other, but they are in closer proximity than before. So these cells are held together by gap junctions, by CAMs, cellular adhesion molecules. They are held together by junctions that allow for the passage of molecules between for the passage of the molecules between the cells. So now we're going to learn about what kind of tissues are there. And we said how many tissues do we have in our body? We have either one of how many? Four tissues. There's either nervous tissue or muscular tissue or epithelial tissue or connective tissue. Those four are very, very, very important. Okay, there are 50 trillion cells of 200 different cell types, and they are divided into epithelial tissue, connective tissue, nervous tissue, and muscular tissue. And an organ is a structure with discrete boundaries that is composed of two or more tissue types. And histology is when we study the anatomy that is not gross. So we have gross anatomy, that is cadaver anatomy, that is anatomy of the naked eye. Yes. It's not there. Oh, I'm sorry. I'm gonna add it. Can you guys follow here, or should I upload it now? Because I began recording, so let's just follow, and then I will upload it to you guys. Okay. Okay, so an organ is a structure with discrete. So five is not there, six and seven are not there, just four. Okay, I will add those. So the organ is a structure with discrete boundaries that is composed of two or more tissue types. And we said histology is to study the microscopic anatomy. And the microscopic anatomy is the study of the tissues and how they are arranged into organs. So a tissue is a group of similar cells and the cell products working together to form a specific role in an organ. So the four primary tissues we mentioned are your epithelial tissue, your connective tissue, your nervous tissue, and your muscular tissue. And they differ from each other in the types and the functions of their cells. The characteristics of the matrix, known as the extracellular material, that is the relative amount of the space occupied by the cells and the matrix. So the matrix is extracellular material that is composed of fibrous proteins, clear gel called ground substance, also known as tissue fluid, extracellular fluid, or interstitial fluid. And that contains water, gases, minerals, nutrients, waste, and hormones. The human development begins with a fertilized egg, which divides and produces first tissue layers. There are three main primary germ layers. The first one is the ectoderm, and that is the outermost layer. And that is the one that gives rise to the epidermis and the nervous system. So these are excellent examples of exam questions. So for example, you'll be asked the epidermis and the nervous system. From which of the three primary germ layers do they arise? Do they arise from the ectoderm, the endoderm, or the mesoderm? And the answer is ectoderm. The endoderm is the inner layer, and it gives rise to the mucous membrane lining the digestive system and the respiratory tract and the digestive glands, among other things. The mesoderm is the middle layer. It becomes a gelatinous tissue that is called the mesenchyme. It is a wispy collagen fiber. These are wispy collagen fibers and fibroblasts in the gel matrix, and they give rise to cartilage, bone, and blood. So interpreting tissue sections. So we have to understand histology. And the main thing that we also have to understand is that we can never understand a particular structure from one way, from one view. So histologists use histological sections. These are medians that to look at on the microscope. The tissue sliced into thin sections, 1 or 2 cells thick. We have two types of sections. There are the fixative sections and the stain sections. The fixative sections, they prevent decay. They fix the tissue in a state that is non-decaying. And you can see that in the formalin that is going to be injected into the cadavers, preventing them from putrefying and breaking down. And there are also stains. These stains color the tissue and they bind to different cellular components. So the sectioning reduces the three-dimensional structure to two-dimensional slice. So that sectioning enables us to examine the structure and the function of the tissue from 3-dimension into 2-dimension. So for example, there is a longitudinal section, This longitudinal section, you would have a tissue, the tissue will be cut on its long axis. In the longitudinal section, the tissue will be cut on its long axis. And there is a cross section, or a transverse section where the tissue is cut perpendicular to the long axis of the organs. There's an oblique section. An oblique section is when the tissue is cut at an angle between the cross and the longitudinal sections. So this is a perfect example of, for example, if we have to look at this egg and examine its structure, if you do not slice it in half, you will never find out that there is yolk inside. You would just think that it's all round and white from the outside. That's why in histology, 3-dimensional is not enough. We have to have a more precise, more specific 2-dimensional structures. So look at the 3 planes of section. There's a longitudinal section. Now you see the structure of the bone and the structure of the blood vessel by cutting it longitudinally. You can cut it also with a cross section. And in a cross section, you see the diameters, they are almost equal, the width and the length. But oblique sections will give you more long than wide, more long sections than wide. Okay, so interpreting the tissue section. So not all tissues are sections. Not all the tissues are sections. Some sections are smeared. So there are smeared and spread tissues. So the smear is a tissue that is rubbed or spread across a slide. A perfect example of that is your blood. So for example, when you want to examine if the patient has malaria, if the malaria parasite is present in blood, you do a blood smear and you examine it. You see if there is any other form of infectious microbe, like a parasite or a virus. Or a virus is too small, you need an electronic microscope, but for that, you need a smear also. And then there's a spread. There's a difference between a smear and a spread. The spread is tissues are laid out. They are fixed. They are put on the slide. They are not smeared and they're not spread. And we just examine the way they are. An example of that would be the areolar tissue. Let's begin now with one of our four major tissue types. These are the epithelial tissue, followed by the connective tissue, followed by muscular and nervous tissue. So the epithelial tissue is a sheet of closely adhering cells. So one or more cells thick with the upper surface exposed to the environment or the internal space. So it covers the body surfaces and lines the body cavities. It constitutes most glands. So if they ask you on the exam, the glands are made up of what kind of tissue? Connective tissue? Are they made up of epithelial tissue, muscular, or nervous tissue? The glands are made up of what kind of tissue? Epithelial tissue. But does that mean the gland is not influenced by the other tissues? Of course not. The other tissues influence one another. So for example, your skin, in your skin, your epidermis, you do have epithelial cells, but these epithelial cells, are they in there by themselves? Is there no blood vessels, blood connective tissue and basement membrane delivering blood to the tissue? Is there no nerve? Are there no nerve cells connected to the epithelial tissue? Is there no muscles that are attached to that skin? Of course there is, but you have to look at the forest as a whole, and then you have to examine every tree that makes the forest. So you have different plants in the forest. The forest is not one type of trees. You have different types of trees that make up the forest, but you have to understand every species of those trees. So in your body, it's like a forest, has different types of trees, different types of tissues, but you have to understand each separate type of tissue. So the epithelial tissue is a sheet of closely adhering cells, one or more cells thick, with upper surface exposed to the environment or the internal space. It covers the body surfaces and lines the body cavities. These are the epithelial tissues, and they constitute most glands. And the epithelial tissue is avascular. It does not have blood vessels. It is avascular, does not have blood vessels, and is usually nourished by the underlying connective tissue. It is nourished by the underlying connective tissue. The functions of the epithelial tissue. What are the functions of the epithelial tissue? Number one is protection. Epithelial tissues are important because they are like provided protection. You know, the money is put in a safe. It's put in a safe. Safe made of iron. You cannot break that. So the epithelial tissue is like a safe that protects the inside of our body. Then you have even more safe, like for example, the bones of the skull. The bones of the skull, they surround our brain. They prevent the brain tissue from damage. They prevent brain tissue from damage so that they are like a helmet. So you're wearing two helmets. If you're driving a motorcycle, you wear a helmet. You're actually wearing two. The first one is your skull, and the second one is your actual helmet. Secretion produce a release. They produce and release mucus, sweat, enzymes, hormones, and other substances. Excretion is voiding these are void waste from the tissue, and absorption, we would absorb chemicals such as nutrients. We would absorb chemicals such as nutrients. Infiltration, all the substances leaving the body are selectively filtered by an epithelium. They are selectively filtered by an epithelium. So the sensation is nerve endings. So in sensation, there are nerve endings in the epithelia that detect the stimuli, because the epithelia, they are the frontier of your body. If you touch something hot, it is those epithelial cells who will first sense that heat before anything else. So the filtration of all substances leaving the body are selectively filtered by an epithelium. So the general structure of the epithelium. So the cells close together. The structure of the epithelial tissue is that these cells are close together. They are small amount of extracellular material. They are avascular. They are avascular. There are no blood vessels, and they are nourished by the underlying connective tissue. There is a high rate of mitosis in the cells near the connective tissue. So the epithelial tissue, they rest on a basement membrane. Epithelial tissue rest on a basement membrane. And there's a layer between an epithelium and an underlying connective tissue. That is your basement membrane. It's a layer upon which your epithelium rests. And they are a layer between the epithelium and the underlying connective tissue. So collagen, glycoproteins, and other protein carbohydrate complexes are available there, and they anchor the epithelium to the connective tissue below it. So these glycoproteins and the collagens and these complexes anchor and connect and cement our epithelium into the connective tissue below it. So now you already know which tissue is immediately in close proximity attaching to the epithelial tissue. That would be your that would be the connective tissue. So the general structure of the epithelium, the surfaces of the epithelial cells is a basal surface. In the basal surface, you have the surface facing the basement membrane, and there's the apical surface, and the surface, it is a surface that faces away from the basement membrane, and there is a lateral surface, which is a surface between the basal and the apical surface, as the sidewall. The epithelia are classified by number of cell layers. These are simple epithelia that have each cell anchored to the basement membrane, and stratified epithelia, and these have cells on top of others, so they do not all contact the basement membrane. So you see, this is a very important diagram that shows us the differences, that shows us the two different types of tissues. There are simple, on the top you have classes of epithelium, on the bottom you have cell shapes. The classes of epithelium, the two main ones are the simple epithelia and the stratified epithelium. The simple epithelium, every single cell is connected to the basement membrane. Every single cell is touching the basement membrane. But the stratified epithelium, some cells are touching the basement membrane while the others are laying on top of each other. And now there's one in the middle, in the gray area. There is the pseudostratified columnar. The pseudostratified columnar epithelium is very important. The pseudostratified columnar epithelium is very important because it shows us that all the cells, they're not 100% one or the other. They're not 100% touching the basement membrane and not folding upon each other. So you see, some cells are touching the basement membrane and touching another cell. Some are touching the basement membrane and touching other cells. Some are just touching the basement membrane. Others are touching the basement and touching another cell. So if you have that mixture with one cell doing both, So here, the cell is touching the basement membrane only and not any other cell. In stratified, these are touching each other and they're not at all touching the basement membrane. These are. But in pseudostratified, you can have one cell who's doing both. It is touching the bottom. It is touching the basement membrane and it's touching the cell. It's touching the other cell. Cell shapes. Cell shapes, you have squamous cell, you have a cuboidal cell, and you have a columnar. You have a columnar cell. Squamous cell, cuboidal cell, and columnar cells. The squamous cell, they are like a sunny side up egg. Sunny side up egg. And there are the cuboidal cells. They are the cuboidal cells, and there are the columnar cells. The squamous cells are like a sunny side up egg, as I mentioned. And the cuboidal cells, they are like a box with the length and the width almost equal. And then columnar, you would have more length than width, like a column. Now we'll talk about the simple epithelium. This one, simple epithelium. So it has a single layer. It has a single layer of cells. It has a single layer of cells. There are 3 types of simple epithelium by the shapes of the cells. So there are simple squamous, because remember, that's the type of the tissue, and these are the cell shapes. So you could be simple squamous, simple cuboidal, simple columnar. You could be stratified squamous, stratified cuboidal, and stratified columnar. It's very important you get this point, that we don't just refer to the tissue by the nature of how the cells are attached to the basement membrane. Here, all the cells are attached to the basement membrane. That's simple epithelium. Here, some cells are attached to the basement membrane and the others are lying on top of each other. But no cell is doing both. So that's stratified epithelium. Here, one cell is doing both. It's laying on top of another cell and it's touching the basement membrane. That's pseudostratified. But that's not enough. That's the type of the tissue. Now, what cell is doing that? You can have a squamous cell being simple. You can have a squamous cell being pseudostratified columnar, squamous cell being stratified. So there's a mixture. It's like a combination. Simple squamous, simple cuboidal, simple columnar. Stratified squamous, stratified cuboidal, stratified columnar. Or pseudostratified columnar. That's squamous. Pseudostratified columnar, cuboidal. Pseudostratified columnar, that's also columnar. That's very important to know. So look at here. That's what we have. 3 types named by the shapes of the cells. There are simple squamous cells. These are thin, scaly cells. These are thin, scaly cells. These are simple cuboidal that are squarch or round cells. And these are simple columnar, which are tall and narrow cells. It's a simple columnar, tall and narrow, simple cuboidal, squash or round cells. And these are simple squamous, thin, scaly cells. But in the end of the day, they all have touching the basement membrane because they're simple. In the 4th type, you have a pseudostratified columnar. Not all the cells reach the surface, but all reach the basement membrane. They falsely appear stratified, as some cells taller than the others. So the goblet cells, this is very important. Goblet cells are wine glass shaped mucus secreting cells in the simple columnar and the pseudostratified epithelium. In the simple columnar, and these are found in simple columnar right here. Simple columnar and pseudostratified epithelium. They are wine glass and they produce mucus. What do the goblet cells do? They produce mucus. That's why you're going to find them all the time where? In the respiratory system, the trachea. the goblet cells. So the simple squamous. So now let's look at simple squamous. Simple squamous right here. Sunny side egg up. Egg sunny side up. What is it? Egg sunny side up or sunny side egg up? Sunny side egg. Okay. Okay, so the simple squamous epithelium is a single row of thin cells, and they permit the rapid diffusion or transport of substances, and it secretes serous fluid, and the locations are the alveoli, the glomeruli, the endothelium, and serosa. Alveoli, the glomeruli, the endothelium, and the serosa. The single layer, this is a single layer of square or round cells. That's the simple cuboidal epithelium. That's a single layer of square or round cells. It's our simple cuboidal right here. Simple cuboidal right here. They permit the rapid diffusion or transport of the substances, and they secrete serous fluid. The locations are the alveoli, the glomeruli, the endothelium, and the serosa. The simple cuboidal epithelium, or the single layer of square or round cells. They are important for absorption and secretion and mucus production and the movement. And the locations are the liver, the thyroid, the mammary, and the salivary gland. So these are all type of, so the goblet cells are simple columnar, and they are found in the simple columnar, and pseudostratified columnar. That's where you find the goblet cells. Now the simple columnar epithelium, these are single row of tall, narrow cells. They have an oval nuclei in the basal half of the cell and the brush border of the microvilli, sometimes ciliated, and may possess goblet cells. There's an absorption and secretion. So the absorption and secretion, they are important for the secretion of mucus. The locations of the simple columnar epithelium are the lining of the GI tract, the uterus, the kidney, and the uterine tubes. So the ciliated pseudostratified columnar epithelium, these look multi-layered, but all the cells touch the basement membrane. The nuclei add several layers. They have cilia and goblet cells, and they secretes and they propel the mucus, and the locations are the respiratory tract and portions of the male urethra. Respiratory tract and portions of the male urethra. So pseudostratified epithelia are stratified epithelia that range from 2 to 20 or more layers of the cell. Some cells rest directly on others, and only the deepest layer attaches to the basement membrane. So there are 3 stratified epithelia and are named for the shapes of their cells. So now we're here, stratified squamous. So now we're here. You see, stratified squamous, stratified cuboidal, stratified columnar. We finished with this. We finished with simple squamous, simple cuboidal, and simple columnar. We finished with that. So the 3 stratified epithelia are named for the shapes of their surface cells. Okay. There's a 4th type known as the urothelium. It is unique to the urinary tract, and they are previously called the transitional epithelium. The stratified epithelia, these are stratified columnar. The stratified columnar is rare, and they're only in places where 2 other epithelia types meet. The stratified squamous is the most widespread, and they are the deepest cells that are cuboidal. They are cuboidal. The deepest cells are cuboidal to columnar, and they include mitotically active stem cells. The daughter cells push toward the surface and they become flatter as they migrate upward. They finally die and flake off, and that is known as exfoliation or desquamation. So a perfect example would be, which type of cells undergo exfoliation from here? The simple epithelium never undergo desquamation. The pseudostratified never undergo desquamation. Who goes through desquamation? See, these are the stratified, and they have many layers because you see they are laying on top of each other. They have many layers, about sometimes 20 layers. So some cells rest directly on each other. So only the deepest layer attaches to the basement membrane. So there are 3 stratified epithelia, and are named for the shapes of their surface cell. They are named for the shapes of their surface cell. You have stratified squamous, stratified cuboidal, and stratified columnar. A 4th type is the urothelium. These are stratified epithelium, and these are only found in the urinary tract. In the past, they used to call them the transitional epithelium. So stratified columnar is rare and is only found in places where 2 other epithelial types meet. You have stratified squamous, that is most widespread, and these are the deepest cells, are cuboidal to the columnar, and they include mitotically active stem cells. So the daughter cells push toward the surface, and they become flatter as they migrate upward. That's stratified squamous. So we're not going to go to the detail of which, if I ever ask you about exfoliation and desquamation, flaking, and the dying off, I'm never going to ask you, is it stratified columnar or stratified squamous? No, I will ask you just the big question. Is it simple columnar or simple stratified? Is it columnar stratified or pseudo stratified? And the answer would be that they are stratified. They are actually stratified squamous, but don't worry about that. Just know it's the stratified. These are the ones who undergo this dying off. There are 2 kinds of stratified squamous, and these are the stratified squamous. You see stratified squamous, stratified squamous. There are 3 types of stratified squamous. 2 types. These are the keratinized cornfield, keratinized cornified squamous epithelium, and these are found on the skin surfaces, and they are abrasion resistant. The non-keratinized, they are found lining the internal spaces, and they lack the surface layer of the dead cells. These are non-keratinized. They are found lining the internal spaces, and they lack surface layer of the dead cells. The keratinized, they are found on the skin surface. So that's why they say keratin is found on the skin, because it is found in the keratinized squamous layer. But on the inside of the hollow organs of your cavities, you would have the non-keratinized squamous epithelium. Keratinized, stratified squamous epithelium. These are multiple cell layers, and the cells become flat and scaly toward the surface. They resist abrasion, they retard water loss through the skin. They resist the penetration by pathogenic organisms. And the locations are the epidermis, the palms, and soles, and the soles are heavily keratinized. So the non-keratinized stratified squamous epithelium. So the same as keratinized epithelium. So these non keratinized, stratified. They are the same as the keratinized epithelium, but without the surface layer of the dead cells. They resist abrasion and penetration of the pathogens, and the location is found in the tongue, the oral mucosa, the esophagus, and the vagina. The stratified cuboidal epithelium is made up of 2 or more cell layers. The surface cells are square or round. They secrete sweat, produce sperm, produce ovarian hormones, and the locations are the sweat gland ducts and the ovarian follicles and the seminiferous tubules. This is the urothelium. That is a multi-layered epithelium with surface cells that change from round to flat when they are stretched. It allows for the filling of the urinary tract, and the locations are the ureter and the bladder. Now we're done with the epithelium tissue. Now we'll talk about the connective tissue. The most important thing you need to know about the connective tissue is that blood is what kind? Blood is what kind of tissue? That is a kind of connective tissue. So in the connective tissue, the most abundant, widely distributed, and variable type of tissue in which the cells occupy less space than the matrix. So of all the different types of tissues, which tissue is the most abundant? The tissue that is the most abundant and widely distributed and variable type of the tissue would be the connective tissue. And most cells are in direct contact with each other. And it shows highly variable vascularity. And there's loose connective tissues, and these loose connective tissue have many blood vessels, and the cartilage has no blood vessels. So functions of the connective tissue. The functions of the connective tissue are to bind the organs, binding of the organs. They connect one bone to another, and they connect to muscle. They connect muscles to bones and skin to the muscle, and they hold the organs in place. So which tissue is highly implicated in connecting bones to tissue, bones to muscle, bone to bone? That sounds like connective tissue, not the epithelial tissue. So for support, to support the body and its organs, they form the internal framework of the organs. These are your connective tissues, and they provide physical protection. They provide physical protection, and they cushion the delicate organs. They protect and they cushion the delicate organs. And they have immune protection. And there are connective tissue cells that attack the foreign invaders. There are connective tissue cells that attack the foreign invaders. Functions of connective tissue. The functions of the connective tissue are, number one, the movement of the bones, and they provide a lever system for the body movement. The cartilages are involved in speech. So the connective tissue is important for movement. It's important for storage. It maintains fat. We will see that adipose tissue, the fat that's being stored in the body, that's an example of connective tissue. It maintains and stores the fat and the calcium and the phosphorus. The connective tissue is also important for heat production, because in heat production, there's a metabolism of brown fat. That generates heat. And there's transportation. So we transport the blood. Blood transports the gases, nutrients, waste, and hormones. So blood is a type of connective tissue. What about fat? Fat and lipid is a type of what kind of tissue? Yeah, adipose tissue is what kind of tissue? Is it epithelial, connective, muscular, or nervous? That's connective tissue. Fibrous connective tissue. Fibrous connective tissue is classified together because fibers are so conspicuous. They are conspicuous components of the fibrous connective tissue. So the cells are fibroblasts that produce fibers and ground substance of the matrix. The macrophages arise from monocytes that phagocytize foreign material and activate the immune system when they sense foreign matter or antigens. There are leukocytes. Leukocytes or white blood cells. These leukocytes or the white blood cells, they function in immune defense. They function in the immune defense. So examples would be the neutrophils attacking bacteria. So this connective tissue is divided into different types. The same way the epithelial tissue, we had simple epithelial, cuboidal epithelial, and pseudostratified. I'm sorry, simple epithelial, squamous epithelial, and pseudostratified epithelial. Then each one is divided furthermore into columnar and cuboidal and squamous. Now, the connective tissue, we don't have squamous connective. We don't have squamous connective tissue. We don't have cuboidal connective tissue. We have fibrous connective tissue. That's the tissue we're going to utilize for healing by scar tissue formation. So when we are injured and we want to heal, not every type of tissue out there can regenerate and enter the cell division cycle and divide to replace the tissue that was damaged. Not every tissue has that property. Some tissues are locked in a terminally differentiated stage. They can never re-enter the cell cycle again. So when they're injured, what do? We just leave it there? No. We replace it with cement. We dump fiber. We dump a lot of connective tissue fiber. So that's known as healing by connective tissue deposition. And these cells known as TGF beta, they will come and they will dump a lot of fiber tissue. So it's classified together because fibers are so conspicuous and components of the fibrous connective tissue are the cells. Fibroblasts produce fibers and ground substance of matrix, and the macrophages. So macrophages arise from monocytes and they phagocytize foreign material and they activate the immune system when they sense foreign matter. That's known as antigens. Foreign matter known as antigens. So these fibrous connective tissue, they are classified together because fibers are so conspicuous, and the components of the fibrous connective tissue are the cells. So in the fibrous connective tissue, you have the fibroblast. These are the TGF beta. These are the ones who secrete fiber tissue. They produce fibers and ground substance of matrix. Macrophages arise from monocytes. Macrophages arise from monocytes. I don't know how. If I go call them, they say one time you want it on, one time you want it off. I don't know. Is it just a button? Things are not always what they seem. Let me see. I'm a professor, I will speak, I will. See, sometimes you gotta you gotta eat your fruits. You see, eating fruits, you get smart. You said that. She said eat fruits, and now she's denying it. That's what the fruit you said you wanted. You know fruits, let me talk about this for a little bit. Fruit has fructose sugar. That is also a type of sugar. So it's not like it's healthy. Fructose. Oh yeah, balance. Balance, moderation. Not too much fruit, not too much Fried chicken. Yeah, that one is bad, but it's good. It's good. It's good as in taste, but it's not good for your health. Especially if you're a professor shouting all the time and you need a lot of blood, and then you clog your own blood vessels with fried chicken. Yeah. Fibroblasts produce fibers and ground substance of the matrix. So macrophages arise from monocytes. They phagocytize foreign material and activate immune system when they sense the foreign matter, known as the antigens. Leukocytes or white blood cells, they function in the immune defense. Examples are neutrophils that attack bacteria, lymphocytes react against bacteria, toxins, and other foreign agents. So plasma cells arise from lymphocytes and synthesize antibodies. So mast cells secrete heparin to inhibit the clotting and the histamine to dilate the blood vessels. While adipocytes are fat cells. These are clustered in some fibrous tissues where they dominate, and these are called adipose tissue. They dominate, and these are called adipose tissue. It's very important. The mast cells, they secrete heparin to inhibit the clotting and histamine to dilate the blood vessel. That's why when you have inflammation, you have a lot of histamine that is secreted. That's why they tell you take antihistamines if you have like some sort of inflammation to decrease the dilation and the excessive blood. And then you have heparin. Heparin is an anticoagulant. It prevents coagulation. So adipocytes are fat cells. They are clustered in some fibrous tissue. So adipocytes are fat cells. They are clustered in some fibrous tissues where they dominate, and they are called adipose tissue. They are called adipose tissue. So these are the type of cells that are found in the fibrous connective tissue. Components of the fibrous connective tissue continue to fibers. So these are collagenous fibers made of collagen, and collagen is a tough, flexible, and strength resistant, and is called white fibers due to the appearance in fresh tissue, and the tendons, the ligaments, and deep layer of the skin are mostly collagen, and they are less visible in the matrix of the cartilage and the bone. Reticular fibers. Reticular fibers are thin collagen fibers. They are coated with glycoprotein, and they form a framework of spleen and lymph nodes, and part the elastic. What happened? So the elastic fibers, these are thinner than the collagenous fibers, and they are made of the protein elastin, and they allow the stretch and stretch and recoil like a rubber band. Reticular fibers, they are thin collagen fibers coated with glycoprotein. They form the framework of spleen and lymph nodes, and they are part of the basement membrane under the epithelium. So collagenous fibers are made of collagen, which is a tough, flexible, and stretch resistant. They are called white fibers due to the appearance in fresh tissue, tendons, ligaments, and deep layer of the skin are mostly collagen. They are less visible in the matrix of cartilage and bone. These are the tendons and the ligaments. Components of the fibrous connective tissue or the ground substance. The ground substance, where is the? So the ground substance is found in the fibrous connective tissue, and it is a featureless substance surrounding the cells, usually gelatinous or rubbery, and it absorbs compressive forces, and it contains large molecules known as glycosaminoglycans, proteoglycans, and glycoproteins. Glycosaminoglycans are long polysaccharides composed of amino sugars and uronic acid. They regulate water and electrolyte balance of the tissues, and examples are chondroitin sulfate, heparin, and hyaluronic acid. These are proteoglycans, and these are gigantic molecules shaped like bottle brushes. They form gravy-like colloids that hold tissues together. So there are adhesive glycoproteins. The adhesive glycoproteins are protein carbohydrate complexes. They bind the components of a tissue together. There are two broad categories. There are loose and dense connective tissue. The loose connective tissue is mostly ground substance in space surrounding the cells. The types are areolar and reticular. Areolar and reticular tissue. They are dense connective tissue and mostly fibrous in space surrounding the cells. The types are dense regular and dense irregular. Loosely organized fibers are abundant blood vessels. They possess all 6 cell types and fiber types. And this tissue underlies the epithelia in the serous membranes between the muscles, the passageways for the nerves, and the blood vessels. There's reticular tissue. The reticular tissue is a mesh of reticular fibers and fibroblasts. It forms supportive stroma framework for lymphatic organs, and it is found in the lymph nodes, the spleen, the thymus, and the bone marrow. Okay, I think this question is on the exam. So this shows you one form of connective tissue, and this is the dense regular connective tissue. It is densely packed, parallel collagen fibers. It is these are compressed fibroblast nuclei, and the elastic tissue forms wavy sheets in some locations, and the tendons attach the muscles to the bones and the ligaments, and they hold the bones together. So densely packed, randomly arranged collagen fibers and few visible cells. It withstand unpredictable stresses. The locations are the deeper layer of the skin, capsules around the organs. Adipose tissue. Adipose tissue is tissue in which the adipocytes are the dominant cell type. Space between the adipocytes is occupied by areolar tissue and reticular tissue and blood capillaries. So fat is the body's primary energy reservoir. The fat is the body's primary energy reservoir, and the quantity of the stored triglyceride and the number of the adipocytes are quite stable in a person. This is the fat tissue. This is the tissue that gives us the energy for ATP, where beta oxidation takes place. Fat is recycled continuously, and new triglycerides synthesized while the old molecules hydrolyzed and released to the blood. Adipose tissue. There are two types in the human. There are white or yellow fat and brown fat. White adipose tissue, known as WAT or white fat, is the most abundant and significant type in the adult. It provides thermal insulation. It cushions the organs such as the eyeballs and the kidneys, and they contribute to body contours like the female breast and the hips. They secrete hormones and they regulate metabolism. Brown adipose tissue. Brown adipose tissue or brown fat is found mainly in fetuses, infants, and children. Adults have smaller deposits of the brown adipose tissue. The color comes from blood vessels, mitochondria, and mitochondrial enzymes. It functions as heat-generating tissue. So the white adipose tissue is empty-looking cells with thin margins, nucleus pressed against the cell membrane. So the nucleus is pressed against the cell membrane. There's energy storage, insulation, cushioning, and brown fat produces the heat. So brown fat is responsible for heat production, and white fat is important for thermal insulation and cushioning and structural support. Then the last of the connective tissues is the cartilage. The cartilage is a stiff connective tissue with flexible, rubbery matrix. It gives the shape of the ear and the tip of the nose and the larynx. The general features are the chondroblast, cartilage cells that produce the matrix. Chondrocytes are cartilage cells that are trapped in lacunae, and the perichondrium is a sheath of dense, irregular connective tissue that surrounds elastic and most hyaline cartilage, not the articular cartilage. It contains a reserve population of chondroblasts that contribute to the cartilage growth throughout life. General features of cartilage. No blood capillaries. When there's no blood capillaries, that's avascular. So the cartilage has no blood capillaries, and the diffusion brings nutrients and removes waste and heals slowly. The matrix rich in glycosaminoglycans and contains collagen fibers. The types of cartilage vary with fiber composition, known as hyaline cartilage, fibrocartilage, and elastic cartilage. Hyaline cartilage. So there's clear glassy appearance because of fineness of the collagen fibers. It eases the joint movement. It holds the airway open and moves the vocal cords growth of the juvenile long, and it's important for the growth of the juvenile long bones. The locations of this cartilage are in the articular cartilage, the costal cartilage, the trachea, the larynx, and the fetal skeleton. There is also elastic cartilage. Elastic cartilage is cartilage containing the abundance of elastic fibers. It is covered with perichondrium. It provides flexible elastic support. It contains external ear and the epiglottis. Cartilage containing large, coarse bundles of collagen fibers. It resists the compression and absorbs the shock. Locations are the pubic symphysis, the menisci, and the intervertebral disc. Bones. The bone is a osseous tissue, is a calcified connective tissue that composes the skeleton. There are two forms of osseous tissue. There is spongy bone and compact dense bone. Spongy bone and contact dense bone is found in delicate slivers. The spongy bone is made up of delicate slivers and plates that give it a spongy appearance. So this bone is a form of calcified connective tissue that we use as mineral and salt reservoir. We use it as a form of a reservoir from which we can deplete salt at any times that we need. So there are two types of osseous tissue. There's the spongy bone and there's the delicate sliver bone. And the plates give it a spongy appearance. So it is found in heads of the long bones and in the middle of the flat bones, such as the sternum. It is a compact, dense bone. It is a denser calcified tissue with no visible spaces. It forms external surfaces of all the bones. The bone features of compact bone. It is arranged in cylinders called osteons. Arranged in cylinders called osteons. The central osteonic canal, it has passages running longitudinally along the bone shaft, containing blood vessels and nerves. It is a concentric lamellae, which is a ring-like layer of bones surrounding the central canal. Is an osteon. An osteon is a central canal and it surrounds the lamellae. The osteocytes are mature bone cells within the lacunae between the lamella. And the canaliculi, these are delicate canals radiating from each lacuna to its neighbors, allowing the osteocytes to contact each other. Periosteum is a tough, fibrous connective tissue covering the whole bone. This is contact bone. Contact bone is made of cylinders that surround the central osteonic canals that run longitudinally through shafts of long bones. So the blood vessels and the nerves travel through the central canal, and the bone matrix is deposited in the concentric lamellae. Blood. So the blood is a fluid connective tissue that travels through the tubular blood vessels. So it transports the cells and dissolved matter from place to place, and it contains formed elements. These formed elements are suspended in a liquid ground substance called the blood plasma. These formed elements include erythrocytes, red blood cells, and they transport oxygen and carbon dioxide. These are leukocytes. Leukocytes are white blood cells. They defend against the infection and disease. These are neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Platelets are cell fragments involved in clotting. Blood smear. So these are erythrocytes that appear as pale pink discs with light centers. Leukocytes are slightly larger and have variously shaped nuclei. Platelets are small cell fragments with no nuclei. Nervous and muscular tissue. Both nervous and muscular tissue are both known to have one thing in common, and that is both of them represent a form of excitable tissue. They have the ability to respond to stimuli by changing the membrane potential. So the nervous and the muscular tissue, they are developed to this excitability is developed to highest degree in the nervous and the muscular tissue. So they are called excitable tissues. The excitation founded in charge differences. So the excitation founded in charge difference voltage across the membrane is called the membrane potential. In nerve cells, changes in voltage result in rapid transmission of signals to other cells. In muscle cells, changes in voltage result in contraction and shortening of the cell. Nervous tissue. Nervous tissue is specialized for communication by electrical and chemical signals. It consists of neurons, and these are nerve cells. Neurons are nerve cells, and they detect a stimuli, and they respond quickly and transmit coded information rapidly to other cells. There's neuroglia. Neuroglia are glial cells that protect and assist the neurons and are housekeepers of the nervous system. They are housekeepers of the nervous system. So these are parts of a neuron. What are the parts of the neuron? So remember that the neuroglia, they are more supportive. They protect and assist the neurons. And these are the housekeepers of the nerve system. Neurons are nerve cells that detect the stimuli and respond quickly and transmit the coded information rapidly to other cells. Parts of the neuron. Parts of the neuron are your cell body, and they house the nucleus and other organelles and it controls the protein synthesis. Dendrites are short branched processes that receive signals from other cells and transmit messages to the cell body. Axons are nerve fibers that send outgoing signals to other cells, and they can be more than a meter long. These are the glial cells, the supportive cells, and these are your neurons. Then we have muscular tissue. Muscular tissue is specialized to contract when stimulated, exerting a physical force on other tissues, organs, or fluids. So it is also an important source of the body heat. There are three types. There's the skeletal, cardiac, and smooth muscular tissue. It is made of muscle fibers that are long, thin cells. These are most skeletal muscles attached to the bone, and they contain multiple nuclei adjacent to the plasma membrane. There are striations alternating dark and light bands. There are voluntary conscious control over the skeletal muscle. They exert voluntary control over the skeletal muscle. Cardiac muscle. Cardiac muscle is limited to the heart wall. The cardiac myocytes are short and branched with one centrally located nucleus. There are intercalated discs that join the cardiomyocytes end to end, and they are striated and involuntary. They are not under the conscious control. There are smooth muscle cells, and these are made of short fusiform shaped cells, and these are cells that have one central nucleus, no striations. They have involuntary function. That is the most is visceral muscle, making up parts of the walls of the hollow organs. Okay, let's take a break. Let us take a break. I'll leave this going on. We will come back. We finish this last part, and then we are done. I'm sorry? Yeah, I'm gonna upload all of that when I'm done. So I'm gonna leave the recording going on. We will take a half-hour break. We'll come back, okay? And then we will finish that last part. Then bye-bye. Go home. Of course, I will do it before you leave. I will do it in front of you. Okay? All right. You guys should have emailed me. I'm gonna put it, okay? I take a break. The recording is going on, so don't. Kamala Harris, Trump. No.
Should compliment you when you do it. Five. Five. Excited for that, yeah. We feel like we can't even see now. You know, with the set, so the machine, and then we have to write anything. Then it was 4 copies, we don't want to say what that. What is it? And she wrote it on the top, and then write 150 more. That's it, right there. I don't think it's worth what, 10 points? Yeah, you guys she said I did. She gave me 0 for one. She did? Yeah. The culture, the one about culture. Oh, the one that we have 10 minutes. Yeah. I kind of know that, but I didn't even know what to do. I kind of know something. Cow, something. She said I did there is a so you give me 0. I told her it's from your book I got it. Last Friday. Last Friday, she's going to discuss with her. Yes. We have class after that. Everybody got that. I think we're done. I tried to do that 20 minutes. 10 minutes to do. 10 minutes, everybody's talking, can't be professor, don't know what's moving forward. She said we're taking her for granted. No, we're not. That was. I'm already doing my homework. I'm already doing my homework. Chapter, week 7. The same question sitting there. So that's too hard. I know, right? It's not too hard. Because you have like you said, she's one back collecting because she don't trust them. It's 2 p.m. What did you do? What did you write? Is that what you do? Just put your own, your own first. But we have 10 minutes. How will it be? I went to copy and paste for that. I didn't. You have to translate in your own words. You can't do that. Yeah, English is not like translating. No, it's not hard either. Something different. You put the other stuff in there. You want to go up to 10 minutes? No, I think you pay tomorrow to do it because of the class. Everything is like I said. The other one I did some is that I did it 28. I said no, you need everything. I'm giving you a homework yet. You know my mic to the by side. All the things like that. I was saying that I was so good. I'm 10 questions you have. Explain very speedy disorder, explain motivation. How will you bring plagiarism? How will copy and paste? Get that on the plagiarism together. Yeah, she said we can even understand we can. When did you do it? What can you add? She only have 5% purchase. Everybody's off. Everybody's off. She said that? In May. She never said that. What did she say? Now we're off. That's why I'm not going to do. You have to translate to everybody. Interesting. She never said that. You said in the beginning, you think well, week 7. Already. That's why she got us last time. You know better stuff about memory. Okay. That's the one we did last week. She didn't go over it. Remember she said, oh, which one does she want to go to 6 weeks. That's the one she still had to begin. That was from week 6. Starts from tomorrow, right? Yeah. For week 7, just in case she passed. So we didn't have time to go over week 6. We didn't do week 6. She skipped it because she wasn't there. She started on Monday. So give us your grade. What did you get? Share the breeze. She took her. I think my time last semester with. I like it also. The more come back, it's was there, like their stuff, you have nothing to do. So you think
it over the egos, and the super ego, that's the more. Ego. Is in conflict, ego. Either way. Yeah.
That's why I did not like Excel.
That we.
I'll not raise it. It's a lot on your own. What you want. The good thing about it is asking you questions. Those are the questions that I put behind the exam to work while I'm at work till 9 times. I will have time to do my homework there and to meeting. And the tests are not pro like this one, if people are watching, the only tests that I like when we get to the question parts. And then I do that, like, yeah, people, you gotta hear you. Well, that's why I went for it, because I feel like it's time to work. Do something fast. Really? And then people hate people last time, I have to forget it, and I can use it. These professionals tell me, you know what I'm saying? I would text the professor, I send them an email, and I would get a response to but it's like, if there is all. It is, it's not easy, literally, right? It's way more writing. See how she's so way more writing for you guys.
Oh, really?
He's lecturing and if you there, so there you have to write. Stephanie is more. More like writing, not type.
But Stephanie is around,
A lot of these topics that she doing. I probably think she does, yeah, no, but we have to put our effort in. So that's discussion, a lot of discussion. And you really have to end comment on certain people. It's their way. Yeah.
Yeah, or sometimes they don't want to market. But yeah, you can get like the you know, they feel it. So they tell you have to take the class, you have to tell people. So next step. But yeah. But you're gonna pass. You're gonna pass.
Yeah, preparing. Listen to her what she said and when she gives you. She's telling you what the problem is. The first question, we don't have much time. Like, let's say when I work Thursday night after, I don't have much time to go over anything. I should have never taken it so early. I should have taken it till, they don't want 9. And yeah, she thinks it's all day. I know. So I should have taken it late, so I can't do the day long, or I have to, after I get a good sleep. But I'm going home exhausted. I have to work tomorrow.
Get up and when you go home, you just take an hour and you just browse through the information.
And it's not for this, that's coming in. The one that's really good. That's why I think that it should be easier. You know, everything in town, you need a sometimes, they go to the life of the person. But after the state of custody, you know what I'm saying? Like one of those things should be on the active, so that's. What's your, it's she embarrassed me, but she does. Yeah, she embarrassed me because I didn't care that it was. She embarrassed me twice, and she didn't, believe you me, she didn't get a third time to embarrass me, because you know you wanna participate. So she would ask the question, and she embarrassed me twice after that, I didn't, I used to not. She embarrassed me. It was not embarrassed, the way like that, it's answered the question like that, the way she, She's not gonna keep you with it. But she, yes, the way she, anybody else who came at me was like, and even, there was a couple of people sitting with us, and they were like, I looked at her like, and when we went outside, they were like, damn, she came at you harsh. I was like, well, whatever. Students like to. What happened? I was gonna think about my autism thing, she's like, no, I'm gonna talk about the internet. I didn't say that, oh, I didn't know, you know. I didn't know that. I was like, oh, that was obvious. Yeah, I don't know. And don't say that. I think it was done. He's like, I don't know. But that's like last year, I was like, I mean, she's like, oh, on the way. Put down on the autism. I don't know that many words. Right? I'm yeah, she's like, I want to take a little piece and look at the diary. Right when I got, I have to start 100 words. And I put, here is the more than 100. She's like, I don't feel it. 200 is on track. And you, she's not saying that. She's counting me? No, I think it's like, it's just a compliment or something. I don't know what they want to do. She really has. So what, you don't get punching off behind me? So what? Oh, God. I gotta work on some stuff she asked you not to write more than what she's asking. Yes, she does. You know. I appreciate that. That's one there, or not even. Give me a mom. I would ask. You want it for it. You got to follow. Oh, she might. Well, we changed me. Well, we can't. You know what happened? They saw how stressed we were, the 6, 7 people that we were sitting in the class, we were so stressed out. And I said, okay, take a workshop. No, we didn't say that. She mentioned. She didn't say nothing. She might have said nothing. She like, I would love to have to suck it up and come to school. I know, we don't even know what to do. Instead of girls. I appreciate her. Still go do Jane myself. No, we got coming too long. Yeah. What do you do, sir? You should put it out to you. We're talking about the communication. I have a couple, I got a big 50. What, for the speaker? When I was there, I don't know how to study that. And she told me to put my name because I couldn't do the country. I'm going to go through and I got one car, which one came up. The other one, no, there was some last night. It was the, huh? Yeah, that's what I heard. Yeah, that's what the, oh, yeah, no, you're right. When the last one? Yeah. Yeah. If you guys ask her actually, you ask him to tell me how it's down tomorrow morning. Bob, where he is? What's the water on that? It's almost 2 hours worth of it. I don't know. Maybe all this time he could have just. I know, he's not finished. And let us go home. I don't know why not said on board. Okay. It's okay, well, I'm not saying that I'm bad. It's not. So if that. We're gonna be talking about you can see that. Right? It's cold. Giving me something I'm. Oh my God. The Friday or the. Is it consciousness or? Yeah. It's consciousness, let me show you where. Yeah, it's on consciousness. You're right. She wants you to know this. This. Yeah. I don't know why. Because it says alpha in stage. They will say alpha for stage one, stage two, instead of like just was like. I think she had to put on that. What stage instead of blah 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 blah. So you're brain, yes, your brain, figures in your brain. You think that there's a child inside, but. Okay, already on.
Hello, guys. I have about 20.
So we have exam tomorrow early morning.
Yeah, I'm gonna finish them. And then when we do the whiteboard, we're gonna explain them even more. So, but we have to cover the slide though. Okay, so now we're gonna talk about the junctions and the glands and the membranes. So cellular junctions are connections. They are connections between two cells, and most cells are anchored to each other or their matrix, and that is through these cellular junctions. So cellular junctions are used to anchor a cell to an adjacent cell or a cell to a matrix. Cells communicate with each other and they resist the mechanical stress and control what moves through these gaps between them. In addition to the cellular junctions, we also have tight junctions. Tight junctions, they are like Z-shaped. They are zipper-like. They are interlocking linkage between two adjacent cells by transmembrane cell adhesion proteins. We talked about those CAMs or CAPs. CAP, cell adhesion protein. CAM, cell adhesion molecule. These are usually hold together between two adjacent cells by transmembrane cell adhesion proteins. They encircle an epithelial cell near its apical surface, and they join the epithelial cell with neighboring cells. They seal off the intercellular space, making it difficult or impossible for substance to pass between the cells. So the example would be the tight junctions in the stomach and the intestines. They prevent digestive juices from seeping between the epithelial cells and digesting the underlying connective tissue. I mean, that's amazingly how tight these tight junctions are. They are so tight in the stomach and the intestine. They don't even allow water. They don't even allow the hydrolytic juice inside your stomach from seeping through to go and digest the neighboring area. Desmosomes. Desmosomes are patches that hold the cells together, like a clothing snap. And they keep the cells from pulling apart. They resist the mechanical stress, and they have a hook-like J-shaped proteins that arise from the cytoskeleton. That is common in the epidermis and the other epithelia. There are hemidesmosomes. Hemidesmosomes or have desmosomes that anchor the basal cells of an epithelium to an underlying basement membrane. And the epithelium cannot easily peel away from underlying tissues. There are gap communicating junctions. These gap communicating junctions, they are channels between the cells, and they are formed by ring-like connexin. The connexin consists of 6 membrane proteins arranged like segments of an orange around a water-filled channel. So the ions, the nutrients, and other small solutes pass and seep through them between the cells. They are located in the cardiac and the smooth muscle, the embryonic tissue, the lens, and the cornea. So these are the tight junctions, and these are the desmosomes. And you see, these are the connexins. So a usual question on the exam is, where do you find the connexin? Do you find it in the gap junctions, the desmosome, or the tight junctions? Structures of the 4 kinds of the cellular junctions. Structures of the 4 kinds of the cellular junctions. This is your intercellular space, and that's the plasma membrane, and these are the cell adhesion proteins or cell adhesion molecules. But in the desmosome, you would have a cell adhesion protein and a plaque and intermediate filaments. That represents a desmosome, where you have the cell adhesion molecule that is not immediately attached to the cell. It's actually hooked to a plaque, and the plaque is hooked to the cytoskeleton. Where connexins are found in the gap junction, and they are permit the passage through this hole. You see, these are the connexins. They allow for the cytoplasmic material, nutrients, ions, like here. Nutrients, ions, small solutes to pass easily between them. Glands. Glands are cell or organs that secrete the substances for use elsewhere in the body, or they or it releases them for elimination from the body. Usually they are composed of epithelial tissue in a connective tissue framework and the capsule. So the secretion is, the product is useful to the body, and in excretion it is useless and harmful to the body. So we have to understand that glands come in 2 shapes. There are endocrine glands and there are exocrine glands. Guys, what's the difference between an endocrine gland and an exocrine gland? Endocrine gland, it secretes its juice into the blood, like insulin. Like the pancreas secrete insulin into the blood when there is too much sugar, or it secretes glucagon into the blood when there is too little sugar. But if you secrete your content as a gland, not into the blood, but rather into a duct, then you are an exocrine gland. Also, like the pancreas secretes some of its content, like these trypsinogens, these are enzymes that break down, the food that's in the digestive system. It does not secrete it in the blood. It secretes it into the duct. So exocrine glands maintain their contact with the surface of the epithelium by way of a duct. Surfaces can be external, like the sweat, tear glands, or internal pancreas. These are salivary glands. The endocrine glands have no ducts, but have many blood capillaries. They secrete hormones directly into the blood. Hormones are chemical messengers that stimulate the cells elsewhere in the body. And examples would be the thyroid, the adrenal, and the pituitary gland. There are unicellular glands. Unicellular glands are secretory glands found in an epithelium that is predominantly non-secretory. They can be exocrine or endocrine glands. Examples are the mucus-secreting goblet cells in the trachea or the endocrine cells of the stomach. So these are the epithelial cells, that's the connective tissue. You see, the exocrine gland will make a duct, will secrete into its content inside a duct. But the endocrine gland, because it secretes its content into the blood capillary, that's why it's connected to the blood capillary. You see, now what is that? Is this an example of an endocrine gland? So in the back, that's the gland. That's the red part. So what does that represent? Endocrine gland or exocrine gland? Jennifer. Excellent. I thought you're in Hawaii, but you're in lecture today. Excellent. Endocrine. Why endocrine? Because the endocrine glands secrete into the blood, but the exocrine secrete into a duct. You see? You see, endocrine glands have no duct, but have many blood capillaries, secrete hormones directly into the blood, and they maintain their contact with the surface of the epithelium by way of a duct. General structure of multicellular exocrine glands. The exocrine glands have a capsule as a connective tissue covering of the exocrine gland. They have a septa or a trabecula, and these are extensions of the capsule that divide the inferior gland into large compartments called the lobes. And there's the stroma. The stroma is connective tissue. You guys said you have an exam tomorrow? You're tired? Okay, go.